Well, Dr. Clark here again. Biology lecture on cell structure and movement. Okay, so today we're really going to talk about how organelles can move through the cell, how the cell itself can move, how materials from outside the cell can get into the cell, how that material can get out of the cell. So we're going to talk a lot about movement and a little bit about structure. So you can watch this little video here. Maybe. And you can see that cell itself was moving, but you can also see, you can see right here in the picture, you can see these ripples, these kind of vein-like structures. Okay, that is support proteins. Okay, scaffolding proteins. And it could be microtubules, it could be actin, but nonetheless, it's some type of protein. Most likely, it's actin filaments that are like when the cell actually hits the little plate there, you can see it kind of fire all these actin filaments at the same time. Now, that's how your muscles work. So an actin-myosin reaction. Okay, so it's probably actin fibers. The remnants here could be microtubules that are still present. It depends on the type of cell and whatnot. Okay, but we'll come back and look at that in much greater detail in a little bit. Okay, so the cytoskeleton is the internal framework. Okay, and the cytoskeleton is really just protein fibers. Okay? Protein fibers that are going to often anchor organelles. Okay, so remember you have chloroplasts and you have mitochondria and lysosomes and Golgi apparatus. So you have all these organelles, but you don't want them all floating to one side of the cell. You want them close to the nucleus. Okay, depending on the purpose of the organelle, you might want them close to the nucleus, so they would be attached to this framework of protein fibers. This is going to be very different than, say, something like a prokaryote. This is for eukaryotes. Prokaryotes might have a little bit of skeleton, cytoskeleton material, okay, but very little compared to a eukaryote. The other thing that it can do is it can allow the cell to flex. So it can allow the cell to compress down and then rebound to the same general shape. Okay? And then it can be extremely important for things like nuclear division. That's where microtubules will come in to play mainly. It can be important for DNA synthesis and transcription translation. So pairing up transfer RNA with the appropriate ribosomes or amino acids with the appropriate transfer RNA, these kind of things, which we'll get to at a later date. Okay. So again, one thing that I want to make sure individuals remember is that when we were talking about the plasma membrane, and it's a snapshot in time, so if I show you a picture of it, I just want you to remember that these are fluid mosaics. Okay? They're movable, and that plasma membrane can move around. Same thing with the cytoskeleton. So just because you might see this internal framework through a picture doesn't mean that that framework doesn't change. It doesn't mean that those protein fibers don't move. We'll see this again, a very good example, when we start looking at mitosis and meiosis and the formation of spindle fibers. And so they're going to come, if we're talking about animal cells, they're going to come out of centrioles, they're going to attach to chromosomes, and they move. Okay, and so they can continuously change. And even the cytoskeleton pieces, the support pieces, can continuously change. So there are really three different types of proteins. I've already mentioned them, but I'll mention them again. Okay, you got microfilaments. This is made of actin. So the protein actin. Microfilaments are going to be a lot of those support and attachments for organelles. Microtubules. Okay, this is protein tubulin. Okay, these are mainly utilized when we're talking about uh, separation of chromosomes, so the copying of DNA and then the dividing of the cell. And then you have intermediate filaments, and these it depends on the cell itself, on how many of these different types of proteins you have and what their purposes are. But a lot of the intermediate filaments are going to be associated with, in some cases, associated with movement, but in other cases, associated with that support skeleton that doesn't allow the cell to collapse completely. So here you can see them. Actin filament, okay, again, it's protein, so it's just amino acids chained together, okay, and these chains wrap around each other. Very important for, movement of material, so something that we call cytoplasmic streaming, okay, which is where organelles will move around the cell. Okay, actin filaments are important for that. Also for the firing of things like the flagellum. Okay, actin filaments are important for that. Microtubules are important for attachment to chromosomes. Okay, and we'll look at these again. And then the intermediate filaments, like I said, kind of provide this support framework. Okay, and sometimes it helps bind things, organelles and things like that, but it also makes it so this cell won't collapse on itself. Okay. So an important piece of the cytoskeleton when we're talking about animals is the organelles, which we call centrioles. Centrioles are the areas at which microtubules will form, okay, and play a really important role in nuclear division. Centrioles also are the place where cells like protista and animal cells that can move, that have the ability to move either by a flagella or cilia. This is the attachment of the tail-like structure. And then I've already talked about their role with microtubules and assembling spindle fibers to attach to chromosomes. So it's pretty important that they're next to the nuclear envelope. The other thing that we're not definite on, but there's pretty good information suggesting that centrioles had their own DNA at one time. And so they probably had a symbiotic relationship or endosymbiotic origin at one time. They've lost their DNA since. Right? They've kind of, but nonetheless, they do show some similarities to prokaryotic functions. So cell movement, again, these protein fibers, these structures are important for that movement. Okay, the actin microfilaments and microtubules can both be associated with movement. Sometimes we got these microfilaments that allow the organism to crawl, and I think some of you have probably seen like amoebas moving, and so they'll extend out a little bit of material and then they'll snake it over. Okay. If you haven't seen this, I'll have a video clip of this when we do the microscope lab. I'll make sure that I get an amoeba and show you that kind of crawling movement of the amoeba. That's an actin myosin firing. Okay? And then sometimes is kind of the beating. So it's going to be an active process, burning up ATP, but the beating of flagella or cilia, which is in coordination with microtubules. So here you can see what I'm talking about for those microtubules. So you have a bunch of microtubules in kind of a sheath, plasma membrane, okay, that's attached to kind of the body of the organism. So a great one that most people have seen videos of it is like sperm swimming. Okay, they swim with a flagellum. Okay, but there are lots of other organisms that do, lots of protista that swim with a bunch of flagella, which we call cilia. Okay, but nonetheless, it's these protein fibers that make up those structures and allow for kind of the movement of that structure. And normally it's either clockwise or counterclockwise. It's kind of a twisting motion in the microtubules, which causes the tail to kind of twirl or beat. I'll show you videos of it later. Okay, so the microtubules have an important role with moving things. Okay, remember when we were talking about the endoplasmic reticulum, the rough ER, making, being the site of protein synthesis, and it's being packaged in vesicles, and then it goes to the Golgi apparatus. Well, it travels along microtubules to get to the Golgi apparatus. And when it gets to the Golgi apparatus, it enters the cis side, exits the trans side, and then travels along microtubules to get to wherever, maybe the plasma membrane, or maybe it's part of the lysosome, okay? Or maybe it's traveling to the mitochondria. Okay? But that it's not, vesicles are not just floating in the cytoplasm or the cytosol. Okay, they're carried, actively carried along these channels, these microtubule channels. Okay. Now, one thing that plants have that animals don't is cell walls, but making up that cell wall or relining that cell wall can be very important. Okay. Other things like fungi and protista have it, but in this class we're mainly going to concentrate on plant cell walls because they're a lot easier to visualize, and I can show you under a microscope and things like that. Okay. And like I said before, when we were talking about prokaryotes versus eukaryotes, and I said that prokaryotes will have a plasma membrane and a cell wall, and eukaryotes will have a plasma membrane and a cell wall. The two cell walls are very different. Okay? The cell walls of plants and fungi and protista are made of chloroplasts, are made of carbohydrates in the form of cellulose. Okay, and most prokaryotic cell walls are made of a different type of sugar. Okay, that makes up that cell wall. Okay? And a lot of times it's going to be accompanied with proteins and things like that. We call it peptoglycan. Okay? But that cell wall is important for maintaining rigidity, and we already kind of talked about this when we were talking about the differences between animals and plants. Animal cells are flexible. Okay? Plant cells are not. They're rigid. Okay? So they don't change shape. Now the internal structure can change shape because they still have a plasma membrane in here. And I'll show you this in lab under a microscope. We can change the shape of the plasma membrane. So it still has the flexibility inside, but the outside flexibility, there's really no flexibility. Okay? So it's maintained shape, and it can grow and be very rigid. And that's what, when you're cutting through wood, or when you chop wood or burn wood, that's what you're burning, is the cellulose. You're burning this outside carbon mat. Okay. Now, some cells, lots of cells actually, have some kind of attachments on the outside of their membrane. So if you remember back when we were talking about transmembrane proteins, these proteins that go all the way through the plasma membrane. Okay? And I said that some of them have this sugar coating on the outside that allows for things to determine if it's self-made. Okay? Even more than that, though, there are a lot of things that can be on the outside of the cell membrane that are attached to proteins. It can be sugar, there can be some other proteins, long amino acid chains, which can have lots of different purposes. Sometimes the purpose is to communicate, like self-made. Okay? So that sugar stamping will be put on by the Golgi apparatus. Sometimes it's to communicate with other cells. Like things can be passed from one cell to the next. Sometimes it's to help with phagocytosis, to bring things in. Okay? So there's lots of situations. But on the flip side, there's also actin filaments on the inside, okay, that kind of also have similar capabilities. It can grab a vesicle, and if the vesicle needs to be moved out, it can help kind of provide a channel for that to move out through a protein. If it's a polar molecule, it has to move through the protein. If it's non-polar, sometimes it'll move right through the membrane. So sometimes it'll kind of give it a little push, move it through the membrane. There's lots of ways at which this can occur. Okay. With that, we'll come back and we'll start discussing energy. That ATP that we're talking about, we'll talk about how it's formed and what its uses are and what what's the purpose of enzymes and things like that in relation to ATP. Okay, till next time.